We are doing a tutorial on DNA extraction from pp2a13.1 and DAR1-1 single and double mutant Arabidopsis thaliana plants, which are very common model genetic organisms. Each of these mutations has light sensitivity, and the pp2a13-1 mutant has a characteristic senescence phenotype in the wintertime day lengths that is capable of being rescued by complementation with a DAR1-1 mutant.
We first collected one leaf from each of the mutant plants we wanted to extract DNA from. We stored the plant tissues in the fridge overnight. We then ground the tissue using a mechanical grinder and metal pellets. We added 500 microliters of CTAB extraction buffer to each sample and incubated the samples for 25 minutes at 55 degrees Celsius. After incubating, we added 500 microliters of chloroform to each sample and vortexed until they appeared smoothie-like. We centrifuged these samples for 5 minutes to separate the DNA-containing aqueous layer from the plant debris. We isolated the aqueous layer in clean, microcentrifuge tubes and mixed the layer with 0.7 volume of isopropanol. We incubated this for 10 minutes at room temperature and then centrifuged the samples again for 25 minutes to pellet the DNA. We washed the pellet in ethanol and dried it for a few hours. Once dried, we resuspended in TE buffer and our isolated DNA was now ready for polymerase chain reaction to amplify our mutant genes.
To genotype our DNA, we made 4 GOTAC PCR master mixes containing chosen gene-specific primers to add to our extracted DNA in order to isolate and amplify each of our pp2a13-1 and DAR1-1 mutant genes separately. We have 4 sets of primers, 2 designed to amplify only the mutant genes, and 2 designed only to amplify the wild-type genes. In each PCR cycle, there are 3 main steps. First, the DNA is denatured into single strands using a high temperature. Then the primers are annealed to each original strand by lowering the temperature to synthesize new complementary strands. The primers guide the Taq polymerase to the mutant gene of interest and extend the DNA from there. The temperature-dependent cycle repeats 45 times. After a few hours of PCRing our DNA, our mutant genes were amplified enough for visualization with gel electrophoresis, since each cycle of the PCR doubles the concentration of the mutant gene.
Gel electrophoresis utilizes the negative charge of the phosphate groups in DNA to move DNA through a gel network using current. We made a 1% cyber-safe agarose gel that creates a network with pores large enough for DNA to travel through. However, the pores are small enough to restrict the speed based on the length of the DNA fragment. Loading and running each of our PCR reactions for about 30 minutes allowed us to detect which strains contain our intended mutations or wild-type DNA. We loaded and ran the PCR products from each of our 4 reactions and imaged our gel. In our image, we see that, for each of our single and double mutant samples aside from our wild-type control, we have bands for our mutant-selecting PCR reactions, and we have no bands for our wild-type gene PCR reactions, signifying the presence of our expected mutation and our success in genotyping.